Along with Sarah Ruhunefard and Meni Wanunu, we've organized this open forum on nanopore sequencing. Welcome to everyone. We'll have a series of talks that are both remote and some presented live here from Northeastern University in Boston. This is the first in a series of open forums in the area of genome technology that the Technology Development Coordinating Center is sponsoring, and look forward to having you at future events as well. Without further ado, I'll turn it over to Andrew Laszlo and Sarah Ruhunefard, who are the organizers and moderators for this session. Welcome to all.
Hi, thank you for joining us for the Pioneer session. I'll be moderating with Andrew Laszlo. So the way this is going to work, we're going to have 20-minute talks from each of our speakers, and then we'll have 5 minutes for Q&A from the live audience. And then at the end of the 4 speakers, we'll have a Q&A that will open up to the both live and virtual. So we're going to start off with David Deamer.
Okay. This is really going to be fun. I'm going to cover about 30 years of history in about the 20 minutes that we have available. And I'm going to cover it in the way of lessons that we have learned as we went through this from an idea to a company that is now producing nanopore sequencing devices. Genomes to proteomes. Next. Can you advance the slide, please?
Just a second.
That's weird. Okay, sorry.
So I give talks like this to graduate classes, and I always like to give them some idea about where an idea comes from, because this is really what any young scientist needs to understand. Next. So this goes back over 30 years to UC Davis when I was on the faculty there. And it's one of those ideas that comes out of basic research. Our goal was to assemble a sort of a model system of artificial cells. Next. Next slide. And I point out that all of us carry ideas around in our heads. And once in a while, these ideas come together in a way that makes sense. And the 4 ideas that I was carrying around in my head were lipid membranes. I knew how to make them. Ion channels. Mark Akeson and I were, in fact, putting the grammacidin channel into planar membranes. Electrophoresis of polyanions moving through gels. And finally, Coulter counters. And those are seemingly disconnected, but they do come together in a way that made sense to me back then. Next slide. So I needed to get ATP into liposomes containing an enzyme called RNA polymerase. Next slide. I might say there's also a mix of people and not just ideas. Mark is now a professor here at UC Santa Cruz. He was a postdoc in my lab at the time, and we were studying the grammacidin channels, so we knew how to make channels in lipid bilayers. Dan Branton was my postdoc advisor when I was at UC Berkeley. He had moved to Harvard by this time, and as you will hear, he visited me in 1991, and that really got us started. I might add that there are other people involved as well. John Kasianowicz, you'll hear about. We just heard his talk. Hagan Bayley is going to talk. Jens Gundlach, I think, is on the schedule. All of these people made a very important part of the progress toward nanopore sequencing. But in 20 minutes, I really have to stick to my own experience, and I'll look forward to hearing their story. Next slide. This shows a liposome with enzymes inside. ATP cannot get across the relatively impermeable lipid bilayer. I thought to myself, well, grammacidin lets stuff in. Why not find a protein channel that can let something as large as ATP across the membrane. There were no protein channels available at the time. This was just an idea. Next. Well, if there is a channel, however, from my planar membrane work, I knew I could put a voltage across the membrane and maybe pull the ATP through into the cell or across a membrane. Then it occurred to me that as the ATP passed through the membrane, perhaps there would be an ionic current blockade. Next slide. This would be a molecular Coulter counter, as Sergei Bezrikov once called it in one of his reviews. Then an idea came along that just popped up and really got me started on this. What if the ATP is not by itself, but instead is a nucleotide in a polymer, such as DNA or RNA? Maybe each base would produce a base-specific change in the ionic current through the cells. So this led me to the first lesson. Next slide. So next lesson is write down your idea. The reason for this is that ideas tend to disappear into mental entropy if you don't write them down. And the second is that these ideas can actually become part of a legal document, as I discovered years later. Now at the lower right, you see an imaginary pore in a membrane. You see an oligonucleotide being pulled through by a voltage, and you see this base-specific ionic current changes. So I pulled off to the side of the road. I was actually driving at the time, wrote this down in what I had to work with, and boy, am I glad that I did that. Next slide. So Dan came to campus 2 years later. He was also interested in single molecule sequencing, and we got into a discussion about this. And this idea of a nanopore related sequencer came up in our conversations, and we decided to go for a patent on this. So Dan went back to Harvard, to the patent office there. We decided to use the Harvard tech transfer office, and they told him that George Church had come up with a similar idea in which a bacteriophage motor would be used to drive a double-stranded DNA across a lipid membrane, hoping to get some sort of information from that. George and Dan and I sat down at the Harvard Faculty Club, and we said to ourselves, we don't know if this idea is going to work. Let's just all sign our names as inventors and see where this goes. Off to the right, you can see the inventors list. Richard Baldarelli was a postdoc with George, and John Kasianowicz played a role that I'll get to just in a moment. Next slide. So we had to patent the idea, and this is where Hagan Bayley comes in, because he was a co-author on a paper that appeared in 1996 in which the actual structure of the hemolysin channel was divulged. It was clear from that that a single strand of DNA can fit through, next slide, and that in fact it nearly filled the channel. Something like 90% of the volume in the channel would be taken up by this strand. Next slide. Alexei Aksamentiev, a little later, I'm sorry that I jumped ahead, that's okay, never mind, made a beautiful molecular dynamics simulation. I just showed that to my classes because that really gives an indication of what can happen. So I asked John Kasianowicz if I could come back to NIST and test this idea with the hemolysin pore that he had available. And all of you know this story. Here's an early experiment that John and I did back in 1993, showing what happens when we put a polyuridelic acid RNA strand, this is a synthetic RNA, into an open channel. The open channel occurs at the top, and of course back then we did not have computational analysis. We had to do this with chart recorders, and we immediately began to see these downward transient blockades as though something was going through the channel. just for fun, I had brought along an RNase, which we put into the medium along with the poly-U, and you can see what happened. The RNase immediately went to work, causing many, many more pieces of the poly-U to be available to go through the channel, and over a period of 10 to 20 minutes, these slowly disappeared as they were chewed up by the RNA. This was really exciting because we realized that we were watching single pieces, fragments of poly-U going through the pore. Next slide. So Mark Akeson joined my lab in 1996, and Dan Branton come back for a sabbatical, and we really got started on this. This is something that Mark put together for our talks, and these are these downward blockades that we began to see on an oscilloscope screen. The next slide shows what it looks like when these blockades occur with an oscilloscope watching this current. And the thing is that these were really whipping through the channel, something like 500,000 bases per second. So one of the main hurdles was to slow this down. Next slide. Meanwhile, Mark had started to work on this question, and we decided there'd be a good idea to see if we could tell the difference with the hemolysin channel between purine and pyrimidine bases. And this was quite something. When we began to see these from a piece of RNA, synthetic RNA with 30 AMPs, 70 CMPs in the molecule, we began to see not just a flat blockade down at the bottom, but in fact, two different current levels, which were obviously being produced by these molecules as they move through the channel. This was really very significant. It helped with the patent application. The patent came out about the same time, and it was published in Biophysical Journal in 1999. Well, there's a lot of skepticism still. Next slide. How could we possibly hope to catch a piece of DNA going through a channel at the rates that we knew it was happening? By the way, we did sort of preview what we thought might be happening, and that little red arrow down at the bottom of this paper that we published in 1996 states our case. With further improvements, the method could in principle provide direct high-speed detection of the sequence of bases in single molecules of DNA or RNA. So as I say, there was a lot of skepticism. We had problems getting our grants approved, but we did get support both from NHGRI and from a couple of commercial places like Agilent and DARPA, and this let us advance the work. Next slide. This shows a paper from a snippet from Mark's 1999 paper, and what I wanted to point out is that this is the first time that the word nanopore had been used in a publication. Next slide. You can see a little square going around the word nanopore, and this of course refers not to the use of the word in a filter. This word was around, but this defined a pore through which a molecule could be traversed under the influence of an applied electrical field. Next slide. Well, the skepticism was somewhat allayed in a 2001 publication with which Winona Vercater, our first grad student working on this, made a beautiful observation. The idea was that we knew the structure of the hemolysin channel, and maybe if we put a hairpin in, the hairpin would get stuck, sort of partly going into the vestibule of the channel, and then would unzip going through the rest of the channel. So our expectation is that we would see two parts of a typical blockade: a partial blockade shown in the center, and then an unzipping and a spike. Next slide. And that's exactly what we saw. Here's a 6 base pair DNA hairpin unzipping in the channel. You can see the open channel current of 120 picoamps going halfway blockade, partial blockade down to 62 picoamps, and then that spike at the end as the thing unzipped. Next slide. The really convincing thing about this is that the order that we could see where we plotted the percent of blockade against the duration that the hairpin spent in the vestibule before unzipping could be predicted from the energy stabilizing the double helical portion in the stem. 3 base pair stem pass through in about 1 millisecond, plus or minus. Then you can see 4 base pairs, 5, 6, 7, and 8. 8 is way up into several minutes waiting around for this thing to unzip. So that really caught people's attention, and I think opened up nanopore sequencing as a distinctly feasible way to study nucleic acids. Next slide. Here is our little gang. We thought that we might have done something important, and that's Winona in the center, Mark, myself, and Hugh Olson, who has been with the group for all of these years and is still working with us in the background. And on the screen, you can see the little slide that I just showed you. Next slide. So lesson 6, license your patents. Gordon Sanghera and Spike Wilcox and Hagan were paying attention to what we were doing, and they founded a company. It was originally called Oxford Nanolabs. It's now Oxford Nanopore Technologies. And Gordon and Spike visited us in 2007 and said that they would like to license patents that we had available, both through the Harvard and through the University of California. So they now have over 1,000 patents, according to what I've read in their publications. And this is a broad intellectual property position. So they wanted to license these. And of course, we said, be our guest, go ahead, let's go for it. Next slide. However, there's a little problem, and that is that hemolysin was not an ideal nanopore. There's something like 10 bases occupying the pore at any given time. We had to find a better nanopore, and this is where Jens really made a contribution. He came to our lab and watched us make the hemolysin nanopore. Then he went home and worked with Michael Niederweiss at University of Alabama, and they came up with the MSPA pore. Jens gave a talk about this at the NHGRI meetings, and it was really stunning to realize that it's going to be possible to find a pore that had a much smaller sensing region and really was going to be able potentially to let us see single bases passing through the channel. However, they were still going through too fast, and that was a major hurdle. That's where Mark Akeson came in. Next slide. Mark went after a motor protein and in fact discovered that several polymerases could act as a motor. He found that the Phi-29 polymerase was ideal for this at the time. As it functioned, it would pull a strand of single strand of DNA through the pore, and this then is on a published in Nature in April 2012. And this also, next slide, was the first time that a piece of DNA had been read by a nanopore. This is a synthetic DNA with the repeating sequence, ACT, 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 and so forth that you see. And this was a real pleasure. It's fun for me to see the next slide, compare this that I had in my imagination from so many years ago. This is very, quite an extraordinary resemblance to what I was dreaming about now come to reality. Next slide. So time to tell others about our results. We went to the Marco Island meeting in 2012 and presented this academic result. But meantime, Oxford Nanopore was making real progress with what they called strand sequencing. Here we are after Clive Brown's talk, at which he introduced the prototype MinION sequencer at this meeting. This was absolutely stunning. It was just amazing to realize that Oxford had succeeded in doing something that we were all dreaming about. Jens is on the left there. A company representative is in the center along with Mark Akeson. I'm saying something to the gang, I guess, and then Dan Branton is down the lower right listening. Next slide. The amazing thing is that Oxford Nanopore, the scientists there did things that we could only dream of. For all of the years, we'd worked with 1 nanopore in a membrane, and it was very hard to get that 1 nanopore situated, in fact. Oxford came up with the MinION, the sensor well, had 2,000 nanopores, and the software chose 500 of these as the best ones to follow. They were individually addressed, and each one was giving recorded signals to the laptop to which this was connected. Next slide. It's always fun to see what can happen with an invention like this. It went up to the International Space Station. There's Kate Rubin floating in microgravity. Next slide. And here she is with a MinION doing nanopore sequencing on the International Space Station in 2016. Next slide. Then we learned some hard lessons. When small companies invent disruptive technologies, big companies sue. Next slide. So Illumina sued Oxford Nanopore for patent infringement. Next slide. Claiming that in fact, Oxford was using the MSPA nanopore, which Illumina had licensed from the University of Washington. This is Jens's MSPA pore. They kind of made this up, tell you the truth. Mark and I were quoted in it, but we did not know what was being used in the Oxford device. They never told us, and we were on the advisory board. So we then were pleased when about a year later, Clive Brown gave a talk. Next slide. And the title of the talk was right out of Monty Python. No thanks, we already have one. He reported that they had a proprietary pore of their own. It's again a bacterial porin, and it works beautifully. Next slide. And this allowed them to escape from the patent suit. They settled it with a payment, and they promised not to use MSPA. They're going to use their proprietary nanopore in all the devices that they put out. Next slide. Another lawsuit. Next slide. PacBio sues Oxford Nanopore for alleged patent infringement. So PacBio had a patent, 9 million and something, and they say that we were infringing on that. This went to a jury trial. And by the way, as advisory members of Oxford Nanopore, our people were involved in this very closely. Next slide. The jury decided in 2020 that the PacBio patents were invalid, and that settled this lawsuit. So Oxford Nanopore has escaped 2 fairly substantial challenges to their nanopore sequencing devices. Next slide. Lesson 12, in the face of adversity, carry on and muddle through. The Brits are good at this. They now have these familiar devices that probably everybody listening has used at one time or another, the MinION, the GridION, the Prometheon. Next slide. There's been a lot of recent progress just in the last year or so. We now know that we can detect epigenetic modifications in a DNA strand as it passes through. Next. Next slide. Accuracy. We now have internal reads of 99% accuracy, and you're going to hear about this as some of these papers get published in the next few months. Next slide. Our reads are now, half of them are over 100 kilobase in lengths. There's even some megabase reads that have been reported. Next slide. These ultra-long reads allowed completion of the human genome. The last 10% has now been added to the human genome by Carol Mega, Evan Eichler, and Adam Philippi were the leaders of the T2T consortium, and they have completed the human genome. Next slide. These ultra-rapid reads allow the record human genome in 5 hours. Just in the last few months, a report from Stanford physicians has shown that the Prometheon was used to get this record-breaking genome. Next slide. And this allowed them to do clinical diagnosis of some of their genetic diseases in patients at the Stanford Hospital. Last slide. And finally, Oxford Nanopore had an IPO that put them over the top with respect to resources, at least in funding. Next slide. I want to read what Gordon Sanger has said about it, because this is where we are now. We are living on the cusp of the genomic era. Our goal is to enable the analysis of anything by anyone, anywhere. Thank you.
Thank you so much for a really awesome talk. I think we're going to hold the questions until the end of the Pioneer session. And up next, I would like to invite Hagan Bayley to join us. He's joining us from the University of Oxford.
All right, yeah, I think I'm all set up. I'm not 100% sure. Maney asked me to give a history talk about nanopores, really against my better judgment. I'd like to be talking to you mainly about new science. But let me have a go. So to go back to Monty Python, this talk is about the very similar to the famous Summarize Proust competition, where you had to summarize the 7 volumes of his autobiography in 5 minutes or less. So I'm going to go right back to the beginning when I started doing science. And this molecule is a hydrophobic photoactivatable reagent, which was used to identify the segments of membrane proteins that are actually in the membrane. So when I started work on all this, there were really no sequences of membrane proteins and certainly no structures of membrane proteins. So going back to the late 1970s, this reagent was used. It partitions into a lipid bilayer, shine light on it, it forms a very reactive carbene, and then that reacts with the part of the protein that's in the membrane, and with a bit of luck, you can identify it. So this shows the state of the art that so many years ago, when we could identify the part of the HLA antigen that spanned the lipid bilayer. So at that time, work on bacteriorhodopsin was also going on. The structure was determined first in 1975, but that was at 7 angstroms resolution. So it looked like this over here. And in 1979, I joined Gobin Khorana's group as a postdoc. And at that time, the first sequence of bacteriorhodopsin was done by his lab and by Ovchinnikov's lab in Russia. And this was the first sequence of a multi-spanning membrane protein. And in 1980, the Cambridge group, Richard Henderson and colleagues, fitted that sequence to this very low-resolution structure, and this is what they came up with. So this was a really exciting time where we were beginning to get sequences of membrane proteins and really understand them at the molecular level. And we were able to show several things. One is it was believed that retinal, which is the chromophore of Bacteriorhodopsin, was attached to lysine 41, using protein chemistry that had been developed for the very insoluble peptides that are generated from membrane proteins. We were actually able to show it was at the other end of the molecule, attached to lysine 216. And also around the same time, we were able to show that you could refold Bacteriorhodopsin. So there'd been a huge amount of work on the refolding of soluble proteins, but we were able to show that membrane proteins were not completely bizarre. They could actually be refolded like soluble proteins if you use the right conditions. And just like amphism, we could show that we could split the protein into separate segments and refold those in a complementary manner. So we could chop off the first 2 helices, refold them with the presence of the final 5 helices, and they would come together and form a protein. So that enabled us to do all sorts of interesting experiments where we could label different halves of the protein with different isotopes and so on. So then I went off to set up my own lab, and that's when I started work on alpha hemolysin. This really goes back to 1985. And in those days, if you took a position as an assistant professor, you had to start a new project. You couldn't continue your postdoc project. And this was the one that I decided to take on. And the basic science question that I was trying to answer was, how can a water-soluble protein assemble into a lipid bilayer? So just as David Deamer emphasized, basic research is really what leads to new technology. And from our early work, we showed that the protein was a hexamer, and there was a hinge region that we thought separated 2 parts of the protein, and it would unfold, exposing a hydrophobic surface that would go into a lipid bilayer. So it's a great way to start as an assistant professor. Both of these things were completely wrong. So, but we went on to correct them. So the problem with using cross-linking to determine how many subunits there are in a protein is that as you get to the higher numbers, 5, 6, 7, and so on, they get closer and closer together on the gel. So we invented a technique called gel shift electrophoresis that later turned out to be very useful for making heteromeric pores. So we could take one subunit that was the wild type, another subunit that was charged in those days by chemical modification, mix them up, separate them on the gel, and we could clearly see that there were 8 bands. And if you just work through this, this means that there are 7 subunits involved. So there's 7 different combinations of the 2 subunits. And at the same time, we were working with Eric Guo to determine the crystal structure of this protein. So this was about 2 years before it came out. And we had a low resolution structure at that time. But by using so-called rotation function, we're also able to tell from the crystals that this protein was a heptamer. And the second thing we did around then, also in the mid-90s, was to find out what part of the protein went into the membrane. And so we did a so-called scanning cysteine mutagenesis, converted many of the residues in the polypeptide chain, so we had individual cysteines in the protein, and we attached a dye called acrylidan that undergoes a blue shift when it goes into the lipid bilayer. And we were able to show that it was actually the hinge region of the protein that goes into the bilayer, so it wasn't the 2 halves that separate and form the contact area with the fatty acids. And also, at that time, we were able to show that this was clearly a beta structure. So if we looked at the blue shift, we changed every single residue to shift to cysteine, and the blue shift went up and down depending on the residue number, as you can see here. So this was clearly a beta strand that went into the lipid bilayer. And also around the same time, again, before the structure came out, knowing that part of the molecule went into the bilayer, we were able to mutate it so it bound metal ions, and this allowed us to arrest the assembly of the pore. So we were able to show that the protein bound to the membrane, 7 subunits came together, they sat on the surface of the membrane like a hockey puck in the presence of zinc, and then when you remove the zinc with EDTA, the protein assembled into the lipid bilayer. So we were getting pretty close to the assembly mechanism before the structural data came out. So around that time, Eric Guo got a high-resolution structure of the protein. He's really an amazing crystallographer. He offered this paper to Nature, but they would only publish it as a short paper, and he was assistant professor as well, and he withdrew the paper from Nature. I mean, I don't think there are many of us that would have actually done that, and then he submitted it to Science, and it was published there. So again, you can see the structure, and one of the primary features is the beta barrel that goes through the membrane, and it is indeed a heptamer. You can see the 7 subunits. And also point out this was a fairly early structure of a membrane protein, if you go to Stephen White's website. And yeah, I think that enabled us really to complete the assembly mechanism. Another thing that we had shown is that if you truncate the pore, so you remove almost all the beta barrel, this will still sit on top of the membrane, and if you apply a potential, it will form a pore in the membrane. And we believe that's because the membrane is actually opening up in this way, and we regard this as a kind of intermediate in assembly. So the assembly mechanism was the protein binds, forms a heptamer, lipid bilayer opens up, and somehow the beta barrel forms. Another paper that's actually more recent that's relevant to this is that this assembly process is really speeded up by receptors on membranes, and on rabbit red blood cells, the protein assembles very rapidly. And on human membranes, it's quite safe to work with this molecule in the lab. It really doesn't work very well. And one thing we were able to do was to attach a ligand, galectin-1, that binds to a sugar on the surface of the human red cell membrane, and show that the protein then assembled very efficiently. So that's actually kind of interesting, but when you look at the structure, I can tell you that this is very far away from the membrane surface once the protein is assembled, and it's still a little bit of a mystery how that happens. So again, during the period when there was no structure of the protein, our main focus in the lab was to find ways that we could trigger and switch the protein, a kind of protein engineering program. And we were able to get variants of the protein that we could turn on with proteases. You've already seen the metal-regulated one, photoactivated proteins, chemically activated proteins. So, for example, we could, by scanning mutagenesis, find residues in the protein that could be modified by nitrobenzyl reagents. Protein would assemble into the membrane, stop at the prepore, can shine light on it, that protecting group comes off, and the protein assembles. So, but once we had the structure, that enabled us to do much more sophisticated protein engineering, and also put the sensing technology that you'll see in a moment on a molecular basis. So we were then able to take the protein, make heteromeric pores that I just mentioned, do normal mutagenesis, site-directed chemical modification, put in unnatural amino acids, replace pieces by native chemical ligation, and as you'll see in a moment, use adapters to modify the binding properties of the pore. So using that, we really went on and developed different ways of sensing ions and small molecules. So one of the first things we did was make a heteromeric pore that had a metal ion binding site in it and binds metals with nanomolar affinity. So you can see here one of our early experiments when we have no zinc around, and then as we got to 200 nanomolar zinc, we get a very strong signal of the zinc just coming on and off the pore. Again, this is a single molecule experiment. And one of the points of this technique of so-called stochastic sensing was that you could detect lots of different molecules simultaneously. So in the case of the metal ions, the same pore could detect cadmium, zinc, cobalt, and so on. And here you can see an example of that where cadmium binds, then zinc, then cobalt, then cadmium, and so on. And you could use this to detect what metal ions are in solution and their concentrations, but it has a kind of eerie premonition of DNA sequencing here, because obviously with the DNA sequencing, you're looking at one base at a time as the single chain of the nucleic acid moves through the pore. So we were able to use that technology to detect a wide variety of reagents, analytes, metal ions, organic molecules, DNA, RNA, peptides, proteins, covalent, even do covalent chemistry. And this work was summarized in 1997 and again 2001. But let me just show you a couple of examples. So here's one. We've made a pore where it will bind a cyclodextrin. Cyclodextrin is shown here. It's just a cyclic oligosaccharide. And if you take beta-cyclodextrin, it has the sevenfold symmetry, the same as the pore, and you can engineer the pore to bind it very tightly. And that enabled us to detect organic molecules, because cyclodextrins are quite hydrophobic inside, and they will bind a wide variety of drug molecules. And again, you could detect these molecules in solution. And here's one of our rather obscure, but actually it's really one of my favorite papers. We found that residue 113 in the beta barrel of the pore, just at the top of the barrel here, really affected how the cyclodextrin bound. And we changed that to every possible of the 20 amino acids, and also to some unnatural amino acids. And we were able to show that actually very few of the amino acids would sustain cyclodextrin binding. And surprisingly, the two of the tightest were asparagine and phenylalanine, which are completely different, obviously. And to cut a long story short, we could show that we could make heteromeric pores if they were all asparagine, the cyclodextrin bound tightly. If they were all phenylalanine, the cyclodextrin bound tightly. But if you had a mixture of those residues, say 4 and 3, the cyclodextrin would hardly bind. And the end of that story was that we'd also determined the crystal structures of these with Eric Guo, and it turns out that the cyclodextrin molecule binds in a different orientation depending whether you've got asparagine or phenylalanine. And that allows you to think about, again, sensing organic molecules in a very rational way. Another thing we did very early on was detect proteins. Now, of course, most proteins won't, unless they're very small, won't go into the pore. I'll come back to proteins again later. And the way we did that was to attach a polymer inside the pore. So again, it's a heteromeric pore. This PEG chain, in this case, has a streptavidin on the end, but you can put antibody on the end, whatever you, sorry, this has a biotin on the end and it's binding streptavidin, but you can bind antibodies as well. And interestingly, when the molecule is bound to the polymer, the very noisy signal you see, it becomes quiet. And without completely understanding that, you can use this kinetic information to sense molecules at nanomolar concentrations. And then finally in this area, we're able to do covalent chemistry. That's a very interesting thing in which we're making a big effort in the lab at the moment. So looking at chemistry in small volumes, if you do this with small molecules, it pretty well reflects the chemistry in solution. But this beta barrel will contain only about 500 water molecules. A single molecule in there actually has a concentration of 100 millimolar. And you can look at all sorts of chemical reactions. I'll just show you one example here that you don't have to understand, but it shows the power of the method. So we have a single cysteine in the protein. It reacts with this arsenic reagent to form two adducts of opposite chirality. So the pore itself it provides a chiral environment. So these are diastereomers, so they're physically different. They will give a different signal. And you can replace this hydroxyl group with different ligands and come up with this very complicated reaction network, 38 possible reactions. But remember, this is a single molecule experiment, so these are all separated in time. And you can measure the rate constants for every one of these. And from a chemist's point of view, this shows that substitution at arsenic occurs with inversion, but it just shows really the power of what you can do with bond-making and bond-breaking reactions. So based on all this stuff, the fact that we could look at small molecules, look at chemistry going on, we formed Oxford Nanopore, as you've heard about from David. So I think if you want a movie recommendation, there's one great movie by Kurosawa called Rashomon that shows an incident that happens, and each observer has a completely different interpretation of what went on. So this is my recollection of forming Oxford Nanopore. It's one of my favorite quotations. Bayley speaks in the laconic baritone of a late-night radio DJ. If he has an entrepreneurial streak, it's not obvious. And yeah, I would say if I had the talent, I'd love to be a late-night radio DJ, but I ended up doing science. And this company was founded in 2005 by me, and then Spike came on, and then we brought on Gordon Sanghera as the CEO, and he's still the CEO. And actually, little known, this was actually a company I founded in 2002, and when I moved to the UK, we bought that out to make Oxford Nanolabs, which eventually became Oxford Nanopore. And when the company was founded originally, it was really founded to detect small molecules, the really biomedical markers. And that Gordon and Spike and I sat around for a long time to decide what markers we were going to look at, because you could detect just about anything by this methodology. But within a year, we decided to take on this much more speculative idea of nucleic acid sequencing. And at the time, we identified, and others identified, several issues. One was getting the DNA to enter the pore for strand sequencing, to identify the bases, to get the DNA to translocate at some reasonable rate, but it actually turns out that not only do you want it to go through at a reasonable rate, you want to ratchet it through the pore, and Jens Gundlach will tell you, I think, more about that later. And then, of course, one pore will take about 2 years to sequence a human genome, so you need arrays of pores. So basically, the academic world was really involved in these first three things at various levels. Of course, they were greatly refined at Oxford Nanopore. And we did some early experiments on base identification in which we could, 2 minutes, give me a few more.
You're eating into your question.
Sorry?
You're eating into your
Good. I like to do that, yeah, because the questions can be difficult. We were able to identify bases in the middle of DNA strands, and we were also involved early on in using polymerases to translocate DNA, and of course this was greatly improved by Akeson and colleagues. And nowadays Oxford Nanopore uses a helicase. Okay, so I definitely want to speed up. So we did quite a lot of work in the lab of developing different ways of recording ion currents, and you can find elements of that if you go and look at the Oxford Nanopore carefully, the chips and droplet interface bilayers. And I think one of the biggest technical breakthroughs that you'll hear about from Lachmal, I think, was developing the flow cell that can have 500 functional pores, but they have a flow cell now that has 3,000 functional pores. And this led to the MinION. I would say that maybe the last thing we did, of course, once Nanopore really got going, we decided in the lab that we better go do something else. But one of the last contributions I think we made was to show that if you had good ways of decoding the signal, you could use more than one reading head to record data. And this has actually come into fruition with a complex of the CSGG pore with a peptide from another subunit, which actually has two reading heads that, again, Lachmal may tell you more about in his talk. All right, let's see. It's according to my timer on here, I'm in good time, but I will speed up. Yeah, okay. So let me just tell you a little bit of new science. So there's a lot going on in the proteome world. There are millions of proteoforms, as you know. Almost 10 years back, we put a thioredoxin protein through the pore using a DNA sequence. So we pulled it into the pore and applied an electrophoretic force. We were able to identify phosphorylated residues at the end of the polypeptide chain by pulling that into the pore, but this is very much a special case. And like many people I can see in the program, we've been developing electroosmosis, which we originally showed for cyclodextrin, that if you generate an electroosmotic force within the pore, it greatly changes the binding and dissociation of neutral molecules. So one thing that we've done recently is chain thioredoxin molecules together. So this is over 1,000 amino acids. And we can pull that molecule into the pore. And again, I have to cut a long story short, but you can see as the 8 different thioredoxin subunits go through the pore, you can basically count them. These are the different length oligomers. And then we've put post-translational modification sites in the middle of this structure. I must say I'm not a great believer in sequencing. I really don't see why you want to, but you know, like Henry Ford, famous last words. So the post-translational modifications, alternative splicing and things I think are much more important. And this is a recent experiment where we've pulled a modified concatenate through the pore, and you can actually see this modification here. This is a glutathione. That's obviously quite a big modification, but the point is that you can see this post-translational modification in the middle of a 1,000 residue polypeptide chain. I'll skip that one, and that jumps forward. Another thing that we've been working on recently is to try and find non-enzymatic ways of moving polymers through pores. And Yuja, who's here, you can talk to her about this in detail, has developed a chemical hopping mechanism where you can move DNA backwards and forwards through a pore using a disulfide interchange reaction that's shown here. So basically this cysteine residue will break this bond, this will move, then the next one will come in and it will move, and so on. And an interesting feature of this is you can control it by controlling the voltage. You can control this from the outside world. So, you can get it to go in one direction and then get it to go in the other direction. And one thing that we'd very much like to do is to transfer this to a surface rather than have it happen in a pore, so we can do much longer sequences of translocations. So I'm going to finish now, because it's now here, it's really come to the my end point. We got multiple times. I just want to say one thing, that engineered pores are very versatile. You can use them for all sorts of things. Some time ago, we developed a method for doing miniaturized bilayer recordings using droplets of a couple of hundred nanoliters, and we could make electrical devices from those, but more importantly, we can print them using a 3D printer, now down to the pico-liter level, and use those to make what we call synthetic tissues. So you don't have to just keep sequencing things. There's lots of other things that you can do with these molecules. So I'm just going to flip to the end and thank all the people in the group, and thank you for listening for just slightly too long. Yeah.
Great. So yeah, up next we have Mark Akeson from University of Santa Cruz. Take it away. We can see.
Great, thank you. Let me play the screen properly. It's brave of you all. In a pioneer session, it's going to be older people who are not very good online, and so it's a brave act to have us do this. My internet, since we're in the middle of Silicon Valley, my internet is unstable. So if I cut out at some point, you can all take a coffee break and so forth. So I'm going to be talking about coupling enzymes to nanopores. I'm a shareholder with Oxford Nanopore and a consultant, so that disclaimer. It's important for me to give credit early on in a talk because I get carried away and towards the end I forget. And some of the foundational studies in the 2000s were, the meetings were in the UK and elsewhere in Europe, in Liège, Ile de Berdère in France, and in Bremen, Germany. And those were foundational meetings for all of us. And likewise, NHGRI, beginning in the early 2000s, championed by Jeff Schloss, was pivotal for our work. And we've been funded by them since the early 2000s for our work. Supported by NHGRI, Oxford Nanopore, and in the early days by Agilent Technologies, who shares some of our fundamental patents, including those that involve using enzymes. And then we got material from New England Biolabs and Enzymatics Corporation that has since been sold, that were pivotal for our work. You've learned about the invention, and these are the people who inspired my work. And this is the experiment that Dave showed earlier, where along with John Castellano, Stan, and Eric Brandon, they executed experiments at NIST and Harvard where they could see polymers, homopolymers of RNA going through the pore and causing downward deflections. And an experiment that Dave didn't talk about, I think, is probably arguably the most important experiment in nanopore history. And that is testing the question about whether or not those deflections were due to actual translocation, as depicted on the right, that the strand is captured in the nanopore and driven through by the electric field, versus on the left, in my mind at the time, when I saw those data, the more probable cause, which was collisions of the polynucleotides with the pore orifice and then diffusing away in the cis compartment. And so that experiment that I think is so critical was performed, and it was decisive. And these are the individuals involved. I think Dan was the one who conceived it, but the hands-on, I believe, were John Kasianowicz and Eric Brandon. And the idea was in the cis compartment, shown depicted here, I think you can see my arrow, let's hope so. They would add either duplex DNA that could not go through the pore in combination with single-stranded DNA that could. And the idea being that you would have regions of these sequences that you could design primers. So if either form of these got through the pore, you could differentiate between, by qPCR, a single strand in the trans compartment or a duplex strand in the trans compartment. And so they ran this experiment. They actually used multiple pores and saw these downward deflections to get enough mass translocated. And asked the question of what went through by quantitative PCR. And the results were striking and convincing for me as a skeptic. In other words, if you amplified material in the cis compartment on the left, you saw both single-stranded DNA and double-stranded DNA. But in the trans compartment, you only saw the single-stranded DNA form. And what's more, those downward deflections they showed were quantitatively, essentially spot on with the qPCR. So translocation actually happened. And Alec Aksmetov later on with his video showed for models of alpha hemolysin that not only did they go through one strand at a time, but the nucleotides were in single file order. So in my opinion, the most important experiment in our field. So this convinced me, I had been a skeptic, but that experiment convinced me to rejoin Dave Deamer in California. And it took us about a year to execute this experiment, which was to see if we could tell between homopolymers of one form of RNA and the other. And as you can see from the graph, based on the time domain and amplitude, we could. And so that was a pivotal experiment. But as others have pointed out, these strands, these were relatively long. These were, several hundred nucleotides at minimum. Each nucleotide transited the pore in about 2 microseconds. And so there were not enough ions, as John pointed out, Kasianowicz, to actually resolve identity of those nucleotides. So the requirement was to figure out a way to slow that. And the person who inspired my work on this came up with the idea, or articulated the idea in the 1990s. That was Dan Bratton, and it shows it left a slide of his of an exonuclease, lambda exonuclease, a depiction of it, what might happen if you digested the complementary strand and allowed a strand of DNA to processively pass through alpha hemolysin. That was for an exonuclease, and on the right was for a polymerase, where the polymerase, as it copied the template strand, would pull the DNA through the nanopore. So that was the inspiration. I'm going to be talking about our work, but we weren't the only ones working on coupling nanopores with enzymes. Reza Gadiri at Salk and Hagan Bayley at Oxford had an NHGRI grant where they also were working on this, and their papers are shown here. I'll be focusing on our work principally. So first up is our colleague Seiko Benner, who's now a research scientist in Japan. And it was sequence-specific detection of individual DNA polymerase complexes in real time using a nanopore, and it was published in Nature Nanotechnology in 2007. For those of you who now use the MinION, this wonderful tiny device that is pioneered and executed by our gifted friends at Oxford Nanopore, you have that tiny device that you can put in your pocket on your bicycle or what have you. And in the day, we basically used massive electrophysiology rigs to do these single molecule experiments that were bespoke for Nanopore. This is an example of the device that Winona Vercotier used for those experiments Dave described from 2001. And so we wanted to focus first on DNA polymerases, and we chose the Klenau fragment of E. coli DNA polymerase 1 for two reasons. One, it was easily available. You could buy it commercially at high concentration. But it's sort of attractive because it was the first polymerase discovered, DNA polymerase, by Arthur Kornberg, who received at Stanford, who received a Nobel Prize for this in the 1950s. So the Klenau fragment of E. coli DNA polymerase 1, how did that do? This shows basically a brief version of the catalytic cycle for this enzyme. In fact, there are many, many steps, some of which we don't know for this cycle. We believe that those are not visible to us yet. But the idea, if you can see my cursor, is this is a depiction of a strand of DNA, the template strand, and a primer strand with a 3' hydroxyl exposed. It binds to KF, the Klenau fragment, to form a binary complex. And then this binary complex.
Mark, are you back with us?
Yes, I'm back.
Okay, go for it. Yeah.
Great, sorry about that. This is going to be an adventure, and I'm going to blame Silicon Valley for this. We take for granted the internet, and then things fail like this.
Just share your screen again.
Yep, here we go. Fortunately, my talk is relatively concise, so we may get through this anyway. So where we were, basically what happens with this experiment, we asked the question if you can see the binary complex. This shows DNA strand per se captured in the nanopore, and the dwell time for this hairpin form of DNA was about 4 milliseconds. If you then added the Klenau fragment, the dwell time increased by about 4-fold, as depicted here. So each of these spots is one translocation. So basically that was binary complexes were visible. And then we asked if the ternary complex could be observed. And this shows a ternary complex, basically the enzyme, the DNA, and a DNTP bound. And for this, we had to solve a basic problem. And that is, if you put all these ingredients into the cis compartment, you would very rapidly fill in all of the strands of DNA. And there would be no experiment to be conducted. And my former graduate student, Jeff Nivala, described these early nanopore experiments as being change of the device. And so if this sort of thing happened to a student who had set up the nanopore and so forth, would be a really very dark day. And so we had to figure out a way to prevent that from happening. And for these early experiments, we were working with static complexes. We didn't actually want to do processivity. We just wanted to see the static complex. And so we borrowed an idea from Fred Sanger, which was to use dideoxy terminators, as depicted here, to prevent catalysis. And so rather than a 3' hydroxyl, it had a hydrogen atom in that position. So static would prevent formation of the phosphodiester bond. And so when we executed that experiment with the ternary complex, Seiko immediately, one of the great things about nanopore is when something works well, you can see on the monitor almost instantaneously. And that's what she observed. The dwell time went from 4 milliseconds to on average 100 milliseconds with the binary complex. And this invites the follow-up experiment that a skeptic would ask. They'd say, well, if that works for DGDP templated by a cytosine nucleotide, then if you took a different templating nucleotide, A, T, or G, you'd expect the complementary DNTP to work. And this is actually a question that Agilent asked us the first time we presented this. And we had executed experiments. So the next slide when we presented to them was to take the different variants, identities in the templating strand. If you took the incorrect DNTP in each case, there would be virtually no effect on the dwell time. But in each case, the correct DNTP would cause this prolongation of residence on the nanopore. So it was working. Advance. There we go. So our goal was to achieve processive strand location. So we had to move on from static models, and we first had to develop a method to measure angstrom scale displacement, a method to activate the enzyme catalysis exclusively at the nanopore, and then an enzyme motor that was retained on the pore under load. And first up was Brett Giarfis in our group, a paper in ACS Nano, and he did a really wonderful piece of work where we basically had a static tape measurer where we could measure displacement at 5 angstrom resolution, or actually better than that. And here's the experiment. We used what are called abasic monomers. So rather than a base at the number 1 carbon on the deoxyribose, there was a hydrogen atom, so basically less volume. And we would then make a trimer of these abasic residues and advance them by 1 position in these static molecules. And you can see that the amplitude of these was substantially different. And when you mapped individual translocations, you could see this sort of staircase. And so we had a way now in alpha hemolysin, which was not sensitive to, as others pointed out, to individual nucleobases. We could see this on the nanopore. All right, so we had a tape measure, and now we needed a way to activate enzyme catalysis at the pore. And there were 2 studies, one led by Noah Wilson, our group, in 2009, and another led by Felix Elazagasti in Nature Nanotechnology in 2010. And here's the basic idea. It's something we called a blocking oligomer designed to prevent enzyme catalysis in bulk phase. And so here is the template strand and the primer strand. This is the n = 0 position where formation of the phosphodiester bond would take place. And what we did was we designed an oligomer, a strand of DNA that annealed to the templating strand and placed a modified nucleobase in the catalytic domain at n = 0. So effectively blocking copying. All right, and so here's the basic idea. At the left, at A, with the blocking oligomer, the enzyme could not bind. In B, if the DNA strand was captured in the nanopore, a positive potential on the trans side, the strand was pulled through the pore and this blocking oligomer was unzipped. We had a way to measure and detect when a duplex was in the vestibule and turn the potential down. And so we could sort of loiter here with the strand in the trans compartment where a complementary oligomer would anneal there. So we've now had this sandwich between a duplex here and a duplex here. We reversed the potential. Now this n = 0 position was exposed. And if you waited long enough, a few milliseconds, an enzyme could bind. We could probe for that enzyme. And if it was present, we could detect its presence. We could reduce the potential and watch replication and movement of the DNA. So that was the basic idea. And we built a map single nucleotide displacements over 12 nucleotides. The basic residues are depicted here. We build a map. So if in fact we were able to get processive copying and translocation, we expected to see sequentially this pattern to emerge from a single enzyme copying the DNA strand. And what we observed instead, unfortunately at the time, this is what we had hoped to see. The ideal scenario is bind the enzyme. DNTP is added, the strand copy is elongated, pyrophosphate is released, and the enzyme advances and cycles, right? So one enzyme doing all the work. See if I can get this to move forward. And unfortunately, this ideal was not observed. The Klinov fragment polymerase dissociated under load. And so here we are reversing the potential, trying to capture an enzyme. And eventually we did, but we would see a single event step rather than a series of events. And then the enzyme would dissociate. Then there would be activity as we reverse potential and see a new retained amplitude, but that too would dissociate and on and on. And so rather than seeing processivity, we were seeing in space individual nucleotides being added and the enzyme dissociating. Let's see if I can get this to work again. And this is basically the model. So rather than seeing processive copying on the nanopore, nucleotides were added in space, and then we would pull the strand into the pore to get it probed again, but then the enzyme would dissociate and we'd have to go back to the beginning. So this was largely, it was good to see, and we had very good control, but we did not get processivity. And we tried another A family polymerase that was reputed to be more processive, T7 DNA polymerase. And what we could see on rare occasion was a couple of nucleotides added while the enzyme was adjacent to the nanopore, as depicted here. So we saw one nucleotide added in a second. But as you might imagine, as gratifying as that was, this was not going to get us anywhere near something that could sequence DNA along a strand. And at that time in 2010, it occurred to us that it was entirely possible this may never work, because we really didn't know what the electric field strength was. Estimates of that had varied by an order of magnitude. And so we moved on to another class of enzyme motor, one that was called.
You've got 2 minutes.
2 minutes? Okay, I'll be done very shortly. Is DNA polymerase from Ph.29 DNA polymerase. And so this enzyme, one of its features is its processivity was substantially greater than the A family polymerases. This enzyme was characterized by the Salas group in Madrid. And what we found was, as anticipated, that the dwell time, here's Klinov fragment, about 4 milliseconds for the binary complex. And for Ph.29 DNA polymerase, we had anticipated a few-fold increase in the dwell time of the enzyme on the nanopore, but in fact, it was thousands of fold greater. So we were on to something. So we went about, we coupled our system with the blocking oligomers to this Ph.29 DNA polymerase. And this video on the right, which I hope will play, shows individual strands being copied or read in one direction, unzipping, and then as it replicated, read in the other direction. See if this will go. There it goes. So this is an individual strand being unzipped. You can see the amplitude differences. And that fast peak was replication in the opposite orientation. And we were able to get on the order of 500 of these read in one experiment robotically. So it's being unzipped, then copied in the opposite polarity. So having achieved this, we formed an alliance with the people at the University of Washington, Jens, who's going to speak momentarily. We put Phe29 DNA polymerase together with MSPA. And this is an experiment. Max Chur from our lab went to Washington, and he and Liz Monroe at Washington put the two systems together. And this image shows a reading of a CAT repeat with that MSPA Phe29 combination. Thank you. So for us, what's next is direct RNA sequencing. We're working on that deliberately, which I think is probably where Nanopore is going to have its biggest impact over time. Thank you.
Next up, we have Jens Gundlach from the University of Washington. Take it away, Jens.
Okay. Alrighty, so this is going to be pretty light history and personal story here in this victory lap session, I guess. Alrighty, so in the year 2000, that's what I was doing. I was doing gravitational research. This is terra firma for me. This is stuff I actually know how to do. And I focused on the most fundamental aspects of physics, really. And so I went with the measurement of the gravitational constant, which was a fabulous measurement. I went to Gila in Colorado, a really nice place where I wanted to have a job. And a friend of mine had set me up there to be on the short list, so the last 3 people that get selected. And what he had not told me was that they were really looking for a biophysicist. I read this job advertisement way too late. And so, but 3 weeks before that, I'd given the talk on the gravitational constant here in this town. And I was paraded through all the labs there, and I talked to Gene Golochenkov, and he introduced me to nanopore sequencing. He was doing this funny stuff. And so I had the guts to tell him at Gila, I'm going to do gravity and I'm going to do nanopore sequencing. Well, I did not get this job, but the bug was kind of in my head. This was one of the slides I presented at Gila. So within the 3 weeks between the visit here to Boston and Gila, I had already realized that one of the problems was that DNA would go through through a nanopore way too fast. And so I came up with this thing here and actually gave a public talk on this. This is a 2-pore setup where you can pull and not push, but you can pull on both ends of the DNA and slow things down. So it was clear to me already that was important, that it was going too fast through there to really read the signals. That's what a physicist sees instantly, I guess. This is what I told him at Boulder I will do. And, now 20 years later, when I look at this, got everything established, actually, including the definite goal, get a foothold in a fast developing field. And you guys all know about the fast developing field. So in 2002, we visited Andre Marzali's lab, and then also Deamer and Akeson's lab. And after that, we set things up in Seattle. And here's our setup. There are parts here that I stole from our teaching lab, another part that Bertil Hille gave me, a kind of vibration isolation table that I took from my gravity lab, alpha hemolysin in here, $200 that I invested of my own cash in a little microscope, and then most importantly, Tom Butler, who was a student in my gravity lab, who was actually excited about taking this crazy thing on. And I owe it to Tom Butler to sit for several years in the corner of a windowless lab somewhere in the bowel of our hospital building. The physics department didn't even want to give me any space in their building for this. And yeah, Tom Butler was really instrumental in getting this to work. We initially worked with alpha hemolysin and even managed to publish some papers on alpha hemolysin. But we knew that alpha hemolysin was not ideal. It had a very long beta barrel here, so which really throttled the ion current. That didn't make sense. And then the constriction that it has was at best mediocre. So we were on the lookout for a better pore. And then in 2004, late 2004, we realized a paper in Science about the pore Mycobacterium smegmatis A. And it had just the right features. It had a very narrow and very short constriction. That is what really turned us on to this thing. And so we sent out, not emails, phone calls at the time. And so we talked to Schultz, who I guess is a crystallographer here, to send us MSPA. And he said, I got tons of this. And he sent us MSPA. And we tried it out, but it did not let DNA through, and it gated a lot. So it looked like translocation signals, but did really not work. Then we talked to Michael Niederweis, sitting here in the audience, who was at the time in the process of moving from Germany to the US. And together we hacked it out and we figured out how to make this pore work. Fortunately, the NHGRI was generous enough to trust us, especially me, who had no background in microbiology really, to support this research. So the problem with MSPA was that the constriction there was rather negatively charged, so DNA just wouldn't go through there. Did not want to go through there. And so we, Michael Niederweis, made mutants to this pore, in which we got rid of the negative charge in the constriction. And since it was so fun, we also introduced a bunch more positive charges at the top of MSPA. But the amazing thing was that these mutants let DNA through, and they didn't gate. One of these things was rational, eliminating the charges. The other thing was just lucky. These mutants just didn't gate as much anymore. So with that, we looked at how these, Jesus, this cuts out all the time here. It's a bad connection in the connector here, with a connector. So we looked at signals from it, and they were rather short, little blips of current reduction. When we used the other mutant that had the vestibule modifications, we saw that there were current reductions, and then at the end there was a blip. And then we plotted them all together, and we found that always at the end there was that blip. So it was likely to be translocation, but not so sure. And then we tried experiments with hairpins, and they stayed in the pore for much longer. But still, we battled because this was all consistent with translocation, but it wasn't really proof. And the worst thing was that PNAS wanted qPCR of the transwell. And that was impossibly hard to do. So it was really impossibly hard to do. So what you do when you're in a bad situation like this, you try something else. So we went fishing. This is a thing that Andre Marzali had done with alpha hemolysin. So you just put, we attach DNA to streptavidin or nutravidin, let the DNA go through, presumably, and then it would, on the other side, catch the fish. So anneal two complementary strands. And then when you pull back, the stuff wouldn't come out instantly. So these fishing experiments worked actually extremely well. And after 1.5 years of our publication being held up by PNAS, we were able to publish this paper, which was Tom's thesis work in the end. And I put all the names here of people involved in that research. So at this point, we had become mainstream nanopore players. And so the problem was still that DNA would go through the pore way too fast. It would go through there definitely faster than 1 base per microsecond, so way too fast. So there were two strategies for slowing DNA down. And the first one was to use duplex DNA. I'll show it to you in a second, what this looks like. And the other one, you heard about it already, is using an enzyme. So strategy one was to take DNA and then expand it with the same piece of DNA between every nucleotide, and then make these pieces double-stranded. And then as this stuff would go into the pore, it would strip off these double-stranded pieces, but then it would wait for a few milliseconds on the original nucleotide, basically, would be held in the pore. So we did these experiments, and this is what we presented in 2009 at the NHGRI conference. And those looked like the holy grail nanopore sequencing current levels. We compared them to homopolymer levels, and sure enough, these levels were exactly indicating the sequence of this thing. We did it at various voltages. The DNA would go through there faster. And anyway, this is a former of DNA that was sequenced within a few milliseconds, 10 milliseconds or so. Whole lot of these traces. We tried it with some separator states. So we inserted other little states which would distinguish between homopolymer sections and so on. Anyway, it worked reasonably well. Something not too dissimilar from what Roche is doing these days, actually. And but it required heavily modified DNA, and the modified DNA becomes very, very long. So not so attractive. We looked for other methods to slow DNA down. And so here we prepared for strategy number 2, to slow DNA down. So here's, so for that we had to show that MSPA really could distinguish between the current levels here really, really well. And here you see there's huge amounts of current between ACGs and Ts. Hagan Bayley actually took the same constriction and showed us that alpha hemolysin really wasn't up to par for this. And so anyway, the currents were large and very well separated, and it was like a perfect ingredient for sequencing. It's another paper by Elizabeth Monroe. We also showed that we would see methylated Cs. And then we looked at the spatial resolution that MSPA really had. And so we hung DNA that had only one single nucleotide modification at position 13, 14, 15, 16, 17, and so on along that DNA that was dangled into the pore. And what we saw was that it didn't really look like it showed a signal for the single nucleotide, but it was spread out over roughly 4 nucleotides. Now, with that, we knew what was the reason for this. It was, mostly KT Brownian motion, so this DNA just moves up and down in the pore and averages out this this current a little bit. But then we teamed up with Mark Akeson here, and we used a polymerase, 529 polymerase, to run it on MSPA, and the rest is history. However, it took us 8 months to learn that detergent-free 529 DNA polymerase ordered from NEB is not detergent-free. But then we saw these traces here. And let's see if I can get this started. These traces here show you these current levels. Obviously, on an oscilloscope, it was just really beautifully how well this worked. Then we went and looked at the data with a bit more detail. And here's this current trace that you just saw on the oscilloscope. Beautifully defined current levels. But what was just amazing is that all these current levels made sense. In this case, the polymerase had let the DNA down first into the pore and then pulled it up later. And there was a beautiful correlation between the current levels as they go in and out. And but what was more amazing was when we repeated this experiment 20 times in a row, all these current levels repeated just beautifully. And not only that, all these current levels corresponded to the DNA that we had put in there. So you can see there's places where there's T's where the current levels are low. Anyway, this was really the demonstration of first sequencing with the nanopore in the hallmark paper by Elizabeth Monroe. So this is, according to the session here, putting it together. This was putting it together. So the MSPA and the polymerase, that is how sequencing worked. We had a great poster that we presented at the AGBT that summarized it all. There's a CAT, the cat DNA that Mark Akeson just referred to. And then we stood in front of the poster. It says here, no photos. And I asked one of the guys to wear the same shirt that he wore then as in the poster here today. Next, we showed direct methylation, hydroxymethylation detection. Andrew Laszlo, sitting in front here, moderating this session, has showed that there's a fantastic current discrimination for CpG methylation, likewise for hydroxymethylation. Then Andrew went on and measured all the quadrumers that I mentioned earlier, so those 4 nucleotide sections, measured them out and did this on long DNA of phi X, of the genome of phi X. And took these data to align Illumina reads to it, so a paper packed full of action, but showing how nanopore sequencing can be done. Then we did other technical advancements. We took a much closer look at these current levels. So here are these current levels that I showed you earlier. The time axis is put into uniform time increments, which is not what the nanopore really does. But anyway, so there's these steps. And what it really is, it's a sampling of an underlying smooth curve that is underlying the data. Here, the enzyme just stops us at full nucleotide positions, but had we stopped the enzyme between these full nucleotide positions, it would have we would have mapped out this smooth curve. And so, if one could only move the DNA back and forth between 2 nucleotides somehow, then you would measure the smooth curve. And we came up with a scheme to do that. And we do this by changing the voltage. So if you change the voltage on the DNA, you can stretch it back and forth between the nucleotides. And so you should be mapping out the smooth curve when you put the data properly together. And so technically, one should be able to see the smooth curve. But when we looked at the smooth curve, it did not look smooth. It had these nasty discontinuities. And so at the beginning, we were thinking, this is terrible. But then we're made out of lemons, we made lemonade. And we figured that that's actually ideal, because now we can repair this by repairing the enzyme errors that are the reason for the non-smooth curve by just setting it up. And when you correct for these enzyme errors, you arrive at the smooth curve, and it helps you to get much better sequencing accuracy. This was shown by in a paper by Matt Noakes and Henry Brinkerhoff, who Henry is in the audience and will give a talk later. But then we also realized that nanopore sequencing had been taken over by industry. For us, that got very uncomfortable. Lawyers and Twitter posts, and it was just not fun. And so what I learned is, when you're on an anvil and they're trying to hammer you, the only way out that you have is go sideways. So we went sideways, and we thought, okay, we can do something else. And instead of straight-out sequencing, we concentrated on the enzymes. And understanding enzymes is a very nice thing to do, because it allows you very precise and mechanistic insights in the function of life. And these enzymes are also drug targets. So if you look at enzymes, and there's a poster back there, where we looked at a very important enzyme that has maybe set in many of your bodies. That's the enzyme that SARS-CoV-2 encodes. And if you can interfere with it, you actually have a possibility to interfere with viral illnesses. So very important to understand these enzymes, these little motors of life, very, very precisely. Coming back to this curve, I'm going to explain you how our single molecule nanopore tweezers method works. So again, there's this underlying smooth curve. So if you were to measure a current level that's somewhere in between two current levels, you'd expect that that the DNA has moved by half a nucleotide. Okay, so we did this kind of thing. So here's the current levels that that we measure with the proper time information to them. Here's a smooth curve. So when you what you can do is you can just map out where every level in this curve here is on the smooth curve, and it translates it for you into position of the DNA. So you do this, and you create a position versus time graph of the where the DNA went. So this is just like in optical tweezers, where people also pull DNA with a force. We apply a force electrostatically in the pore, and the enzyme is anchored somewhere, and our enzyme rests on top of the pore. So the amazing thing was, if you look at it, then we saw here that this enzyme moved at steps of 300 picometers. But these 300 picometers are resolvable to some incredible precision. You see how little noise there is on a step. So this made us realize that this is a fantastically sensitive single molecule method to analyze enzymes. It has sub-angstrom position resolution, sub-millisecond time resolution, and at the same time, it gives you the sequence that went through the enzyme. So much more powerful than optical tweezers. Here's such a data trace taken with a helicase, HELT38. And what you can see is this thing moves at half nucleotide steps. So there's between nucleotides, between every nucleotide, there is a step at half the distance that it traveled from nucleotide to nucleotide. And then when you study just the duration that it spends on every level here, you can even deduce from it that this helicase not only went in 2 steps per nucleotide, but that these substeps have substeps in them. And so my graduate student at the time, now postdoc, John Craig, made this fantastic reaction diagram of what the helicase does, and really figured this, put us mainstream into enzymology to figure things out. He looked at one thing that I thought was this arrow mode, where it's this backstepping. But the backstepping was actually super interesting when you correlate it to the sequence in the enzyme, because the backstepping is strongly sequence dependent. So here, for example, you can see that whenever it is on a C, then the probability of backsteps is very low. So he went, and this is, by the way, something that you can actually now use for sequencing again. You use the kinetic information. So this is a totally independent readhead by using the time information, the backstepping information, for example, to identify if a nucleotide that went through the enzyme is a C or an A. Okay, time is running really short here. Let me just show you some data of an RNA polymerase. And the RNA polymerase makes these long pause states. And interestingly enough, while it usually steps from full nucleotide to full nucleotide, in the pause state, it ends up at a half nucleotide position. But when you look at it with our method here, you can see that at the very end, it does go to the full nucleotide position and then moves on. That just shows you that this position resolution that you can obtain with nanopores really helps you to decipher how these enzymes actually work in reality. We have lots of collaborations, and let me just more or less end up here with explaining you why this thing is so sensitive. On the left is optical tweezer data. On the right is Sprint. You can see that it's the same enzyme. You can see that Sprint has fantastically, or the nanopore method has fantastic resolution, while in optical tweezers you can just barely see this. The reason for this is, you can see that here. And this thing here is not a speck of dirt. This is the nanopore setup. It's much smaller than optical tweezer setup. And the reason why the optical tweezers don't work so well is not because you can't figure out where the position is of the beads in optical tweezers, but because there has to be a long tether in between that's just springy and flexible and does not allow you to do the same thing as we do. So to drive it home more, here's this whole thing magnified out. This is the length of the tether that we have for a nanopore setup, and the other bead of the optical tweezer setup would be somewhere outside this building. Okay, with that, I will just conclude. It's not really a conclusion because we're actually moving on. There's no conclusion here. We're moving on. My group has been at the nexus of nanopore sequencing. Collaborations and team efforts have made this possible, and there's never a dull moment in the lab. So with that, I'm done. Okay.
So we're going to move on to a sort of group question session with all of the speakers from this session. So Jens, I'll have you stay up here with this mic. Hagan, if we could have you come up with this mic over here. And then Mark and David, are you guys still with us?
Yes, I am.
Okay. So yeah, we'll open up the floor to questions if anybody has any. Yeah. And when you ask a question, go ahead and say your name and where you're from as well. So
Jason Dwyer from the University of Rhode Island. This is a question, I think, for everyone because you've all done magnificent things by changing protein pores, but I'm going to start sort of targeting Hagan for a minute, if I could. You've done some really intriguing work outside of just sequencing, and in particular, if you look at the cyclodextrin, that is itself a pore. And have you ever thought about using fully synthetic pores as a platform?
Well, we've thought about it. You know, as there are many people using solid state pores, for example, and we've had to go, depends what you mean by synthetic, we've used DNA nanostructures to order alpha helices and so on and form pores in that way. But I'm still a big fan of protein nanopores because you can engineer them really at sort of angstrom resolution, which is really not possible with solid state pores, and I think not quite possible with synthetic pores made from peptides, but probably will be soon.
Yes, it will be.
Right. I'm actually meaning small molecule organic frameworks, so stuff like Stefan Matilli uses. So where
Yeah,
you have a
there's
where you
been a huge amount of work in that area. But again, I think very often the structures of those molecules are really not known, which is a bit of a disadvantage. And again, it's much harder to engineer functional groups into them at specific positions. So yeah, I'm still favoring protein pores.
We actually have a collaboration with David Baker where we look at totally synthetic pores, but they're protein pores.
Yeah, Jens, I guess they're still made of alpha amino acids, right?
Of course.
So, yeah.
But they're pores that had not been seen in nature.
Yeah.
Nikolai Slavov from Northeastern University. Collectively, you tell a wonderful story of a dream come true with a lot of progress and triumphs. But I wonder if there were at times periods when you feel progress could have happened much faster than it actually did. And if you can look back and see at opportunities to further accelerate the development of nanopores, were there some things that you wished had happened differently?
Yes, of course. In hindsight, everything is easier, right? I mentioned a few things where our publications were held up for a year and a half, and I think that slowed down the dissemination of results by a lot. So there are those times.
Yeah, I can't really answer that, but I would say that a sense of humor is one of the most vital things in doing science.
I want to add to that. With students, there was a lot of skepticism, and I think Dan Bratton pointed that out in an editorial at one point. And I try to convince students to be, and I was a skeptic of nanopore at the start, to be honest with you, but to be an open-minded skeptic, right? You need to bring both things to bear. And fortunately, people like Jeff Schloss had that quality about them. And so fortunately, there were people who persevered and helped support this. And that, I think that sort of hard-boiled skepticism delayed things a little bit. But this kind of thing, I think, typically, if you look like at AI, for example, there was huge skepticism there too, and people said it would never work, and it took decades to break through as technology advanced. So I think, I can't imagine that this thing could have been sped up much more than it was. Fortunately, there was the idea and some people who persevered.
I think that when Oxford Nanopore finally got started, it really showed how much of a difference it makes to have 100 million pounds instead of, you know, $1 million, and 100 employees as opposed to 2 or 3 grad students and a postdoc. Mark and I, in fact, Mark has, I'll quote him, it was like hanging on to the tail end of a rocket when Oxford Nanopores finally got underway. And even with all of that, it still took, 10 years? Well, not quite 10 years, say 7 or 8 years to get all the way through to the first marketable MinION that was put out in 2014. So just money and people, that is a impediment if we really look at it from this perspective.
But I think, David, you'd probably agree that the basic science, the underlying science, very often comes from academia, you know, and it has to be taken to a certain level before a company can run with it. One fear I have with the way in which funding is happening nowadays, with the exception of the programs that we've been in, thank goodness, and the pressures that are put on grad students and postdocs, that it's getting harder and harder to take on more speculative science projects, and I think that's what we should be doing at universities, but it's getting harder and harder to do, particularly for new assistant professors.
Yeah, sometimes you just got to take a risk. I certainly did that. Yeah, I know. Tom Perkins. He does optical tweezers.
But I must say, I recall one great lecture I went to by Dave Deamer, and he'd gone out into the Kamchatka Peninsula and put some lipids into a hot spring. And we were all looking at this, we were really intrigued, and he turned to the audience and said, I wouldn't do this unless you've got tenure.
Yeah, that's right. By the way, in about 15 minutes, I'm giving another Zoom talk to an Israeli audience. And the slides of that experiment really gave us an idea that we're still following up. And we're using the MinION, by the way, right now to try to demonstrate non-enzymatic sequence transmission from a template strand to a product strand under the conditions that we think prevailed on the early Earth. So I'm kind of coming full circle back to this problem of using nanopore sequencing to approach one of the most difficult problems, and that is how life began, where nucleic acids really came from early in the history of the Earth and of life on the Earth 4 billion years ago. And the question is, did nucleic acids pre-exist, and were they sort of caught up in this system that we now call a living system? Or did somehow life come to be and then invent nucleic acids? And that's a valid question. And my guess right now is that there are mechanisms by which nucleic acids can be synthesized just from the nucleotides without enzymatic activity. And that these are random sequences which then are subjected to evolutionary pressure and finally got encapsulated into protocells, which basically is what I was trying to make back in 1989. We were trying to put together an enzymatic system inside lipid membranes that would have some of the properties of life, such as synthesizing RNA by an enzymatic process.
Okay, we've got time for one last question.
Very nice talk, to listen all of the top nanopore experts. And I have one question for Hagan, because you mentioned during Oxford Nanopore, it starts with diagnosis. So in my personal understanding, sensing some biomolecular should be much easier to do the sequence. You can use all the parameters rather than only current to define ATCG. So what's the reason behind, or why sensing like sequences become the main goal rather than the sensing?
Yeah, originally we viewed nanopores as a platform technology. So basically anything that's in an aqueous environment can be sensed by nanopore technology, whether it's an ion or small molecule, all the way through to polymers that we're looking at nowadays. But when we started the company and got funding for the company, we were definitely focused on small molecules. Although when we gave our talks to these investors, we always mentioned the possibility of sequencing on the last slide, and they always look sort of vaguely interested. But when we started the company, I think we were fortunate in those days because you could get so-called patient capital. So the investors weren't looking for a return in 3 months or 6 months. They were willing to wait 5 years or maybe 10 years. So we decided at a certain point to take on this sort of bigger challenge, if that answers your question.
So if I understand right, it's about the funding, not about the fundamental behind it, like the diagnosis, the complex matrix is more challenge for sensing, something like this.
Yeah, I don't know. I think with Oxford Nanopore, it lays somewhere between academic science and, company that's trying to churn out a new bottle top or something. And so it was definitely a sort of research project to begin with. And I think the art of doing good research is to choose something that's barely feasible and do it, right? And I think we thought that small molecules were pretty mundane target, and also ultimately would not make lots of money from the company's point of view, but also from being important in medicine. Obviously the sequencing would be revolutionary.
So we have one final question, just a quick one before we go to lunch, from our online audience. Will it be possible to do nanopore sequencing without motor proteins, for example, using electronic controls?
Well, I think ultimately, yes, it will be possible. The idea is to engineer the functionality of these enzymes basically straight into the nanopore, and it's all one and the same. And the machinery is is inside the nanopore. I mean, some of these enzymes that we had been using, we used them so poorly, in such a poor fashion, where we, for example, in the Phi-29, we just pulled the DNA through there, not even in the direction that the enzyme wanted to go. So all we used were these holding modalities that are inside the enzyme to, it was just a break. It was, so you don't need the sophistication of the enzyme. And I think Hagan also alluded to this with the, you know, the hopper thing. So, yeah, ultimately, it will probably work without an enzyme.
Well, I think we're eating into lunch now. I see a lot of hungry faces out there and would rather be eating lunch than eating into lunchtime. So with that, let's thank these speakers one more time.